Okay. All right, bulk transport. So sometimes molecules are too big to go through the membrane. They're bulky. So the lab has an error. The lab says that bulk transport is to move things that are too small through the membrane, which doesn't even make sense. How is it possible for something to be too small to fit through the membrane? So what it should say there is that it's for things that are too big. So if you remember when we were talking about simple diffusion, here's sort of a section of the cell membrane. In simple diffusion, you had a situation where something was small enough that it literally could fit through or in between the phospholipids. And that only applied to really like oxygen, carbon dioxide, and very small hydrophobic things. Then yesterday, this is near the end. Yesterday we talked about facilitated diffusion and active transport. In facilitated diffusion, it moved bigger things that couldn't go through the phospholipid bilayer from high to low, but instead of going through the phospholipids, it went through protein carriers. And then active transport carried things from low to high, also requiring energy and using protein carriers. Bulk transport is for really big things. So if you imagine that I have some molecule and it's this size, this is too big to go through the protein, it's too big to go through the phospholipid bilayer, so the only way that a cell is going to be able to take it in would be bulk transport. In general, there's really one word for bulk transport taking in, and that's endocytosis. Like this, where a cell literally folds in, it surrounds and engulfs a particle, and then the particle ends up in a vesicle inside the cell. And exocytosis is the opposite of this, where the cell spits out a particle. So in essence, that's what bulk transport is. It's where a cell literally has to surround and eat something large. All right. So endocytosis is a type of active transport where a cell surrounds and engulfs a molecule. It can be subdivided into three, subdivisions of endocytosis. So and this is really based on the particle figures. He's been quiet all day. It depends on what kind of particle it is. If it's a really big particle, then the cell would take it in by what's called phagocytosis, which we mentioned in the previous chapter. Like your white blood cells eat bacteria by phagocytosis. Penocytosis is where the cell takes in a large group of little particles all at one time. So it would look like this. They might be small enough that they could maybe pass through the membrane another way, but this would allow a cell to take a large group of them at once. Sometimes this is called cell drinking, or that's what it would translate to. Penocytosis, a bunch of small particles, but all at one time. And those small particles would then end up in a vesicle, like this. Now they're trapped in a vesicle, a piece of the membrane that is broken off, and then that could go, for example, to the lysosomes to be digested. The last one is called receptor mediated endocytosis. And this is where you have a cell, and the cell has protein receptors on the outside, and the receptors, when they bind to some particle, that stimulates the cell to absorb the particle. So here's the cell and the receptor, here's the particle, and basically the binding of the particle to the receptor told the cell to eat it. So that's called receptor mediated endocytosis. So again, all three of these are subdivisions of endocytosis, and in the Quizlet, I made a Quizlet of pictures where you match the parts to what they look like, just so you know. And again, the opposite of endocytosis is exocytosis. So cells take in particles through endocytosis, cells get rid of particles through exocytosis. All of these are, again, what we call bulk transport. The cell is surrounding and engulfing a large particle or a large group of particles, something that's typically too big to go straight through the cell membrane. Any questions on that? Yes? Yes, I know there's one more word, and you actually won't be tested on it, but I will give you that word. Let me show you, before, I give you that last word. Here's a diagram, kind of comparing the three. So this is phagocytosis on the left. Notice it's a really big particle. Penocytosis, it's a bunch of small particles at one time. And this is receptor mediated. The receptors are on the surface. The cell, when the molecule binds to the receptor, that tells the cell to go ahead and surround and engulf the particle. So it sort of sends a signal to eat that particle. The word you're looking for is hypercholesterolemia. You won't be tested on it though.